So as Mike said, my name's Becky McClellan. I'm a genetic counselor. I've been actually practicing here at Kennedy Krieger for about 16 years. I now, as you see on some of my slides, split my time between the Johns Hopkins Center for Inherited Cardiac Disease as well as Kennedy Krieger. So I do a little bit of back and forth. But some of that change has actually introduced me to a lot of some of the newer tools and kept me on my toes as far as genetic variations and understanding what they mean. And so that's, I think, part of what I've been asked to share with you and to talk a little bit, not only about a little more about the basic genetics that Dr. Fatimi started to introduce this morning, but to talk a little bit about what it means to have a genetic variant within the DARS2 gene. And we'll talk about some of these things like splice mutations and frameshifts and all of that. And I will do my best to bring it down and make it easy to understand. All right. So just really quickly, I think, because you can never hear this too many times in different ways to help it sink in, right, is basically a gene is a working unit of DNA, and the genes tell the body how to make all of those different proteins. So going back to the things Dr. Fatimi already introduced, that DNA code is basically made up of 4 nucleotides or base pairs. We have lots of different terms, all meaning the same thing sometimes, just to make it a little easier to follow. But typically we represent them as A, G, T, and C. And they are basically the code that forms our DNA, and then functional or working groups of those DNA letters are what we refer to as a gene. All of that is packaged up in a tiny little way into the center of all of our cells in the nucleus of all of those cells. So a little more genetics 101. So the nucleotides are essentially those DNA base pairs of the codes that make our amino acids, which then turn into the protein. So Dr. Fatimi talked a little bit about this morning as well. So this first step here is basically showing you the DNA letters, which get turned into their copy, right? Even though they don't look quite identical, they are a copy. And then every 3 letters of those DNA basically form an amino acid, which gets strung together like beads on a chain to make up the protein. This guy over here is kind of a fancy picture that actually shows you the code, just like Da Vinci's code. So every 3 letters of those DNA will tell the body to bring in that certain amino acid. So that tRNA sort of matching that Dr. Fatimi was talking about before, we only have so many different amino acids, but we have 3 billion DNA letters that make us all who we are. So there has to be a system that kind of tells every certain letters to kind of turn into what they're going to turn into. And so this is sort of showing you that, if I lost my little pointer, but it's showing you the center. If you work in the center and work out your first letter, your next letter, and then it gives you the amino acid on the end. So it's just a visual way to see what those DNA letters, how they turn into the different protein. But DNA, it's just hard, right? Because there's a lot that has to be remembered, and we know that this is a complex topic. So one of the specific questions, again, Beth has been wonderful, I think, with feeding us things that you have asked and making sure that we are trying to address the concerns and questions. And one thing I think that comes up, and Dr. Fatimi touched on this morning, is this idea of DARS, LBSL is thought of as a mitochondrial disease, right? But is it actually caused by a change in the mitochondrial DNA, or sort of the difference between what we call the mitochondrial DNA and the nuclear DNA? So in our cells, we basically have, we'll go to this. We basically have a nucleus, right? That's the center of all of our cells. The nucleus has 3 billion DNA letters that make up about 20,000 different genes. DARS2 lives in the actual nuclear DNA, and that's important when it comes to recurrence risk and inheritance and how this condition is passed. We also have 37 genes that live inside every mitochondria that are in our cells. And in actuality, a lot of our slides today, we've shown it looks like each cell just has like one. Your cells actually have lots of mitochondria in them, so there's multiple sets of that mitochondrial DNA floating around in each cell. It just depends on how much energy that cell needs, and then the cells that need more energy are going to have a whole lot more mitochondria to help make it. But so there's mitochondrial DNA here, 37 genes, and then there are 20,000 genes that live in the nuclear DNA. That's where DARS2 lives. Both changes here as well as changes here can cause mitochondrial disorders, meaning disorders that affect the way that the mitochondria work and provide function for the cells, right? And so we will often refer to these. You'll see genetic conditions will have an MT before the gene name, or a mitochondrial DNA will differentiate where that gene is. But we know that there are probably several hundred to 1,000 genes that live in the nuclear DNA that help provide mitochondrial function and support. So you can have changes on either side that can end up as a mitochondrial disease, but they're all different and are caused by different genes that might live in one or the other place. Okay. Again, so basically a mutation, you'll hear that word. I tend to prefer to use variant, which is the term that we hear a lot of us use as well. It means the same thing, right? Variant has a little bit of a more positive connotation than a mutation, right? Teenage Mutant Ninja Turtles got really popular, and so then we kind of stopped using that word. But basically, there are three primary ways to cause a change in the gene. And the first is a misspelling. So if the DNA code we've talked about needs to be in a very specific order and combination, let's say it has to go GATG in that order. A misspelling is something as simple as instead of being GATC, it's GTTG. So one letter gets switched out for another. We'll refer to that as a misspelling. You can then also have extra DNA added in or missing DNA, and that can sometimes be one DNA letter or sometimes it could be multiple DNA letters. And we're going to get into some of the more specific types of genetic changes a little bit later in the talk, but those are kind of the basic foundations for how a DNA can get changed and potentially lead to a genetic condition. Inheritance, right? So you have 2 copies of every gene, one you get from mom, one you get from dad, right? Some genetic conditions, if you have a change in just one of the 2, that's enough to cause disease. Other genetic conditions, you need to have changes in both of them. I just did it myself, sorry. Both of them in order to present and cause disease. We refer to that as autism or recessive inheritance, and that's the inheritance pattern that LBSL follows. So you need to have 2 changes affecting both copies of the gene in order to present with disease. And the other thing to know is that even though something is genetic, it may not always be something that came from both parents. All genetic changes have to start new somewhere, and that may be that both parents have a change and it got passed to the child, or one of the parents has a change and then the other parent didn't, but the child got a de novo or a sporadically new change. So often both parents are carriers, meaning they have one change in LBSL, but it is not always the rule. Okay. So this is just a picture that kind of shows the outcomes in an autosomal recessive condition. And again, in this image, we are using the presumption that both parents are carriers. And I think you guys all know this, and so I'm not going to go through it in a lot of detail, but happy to ask and answer personal questions about your cases if you have questions. So LBSL, they asked me to include just a little bit about the history. Dr. Fatimi touched on this earlier, but essentially it first was published in the early part of 2003, where it was first clinically described in 8 patients by Dr. Vandernaap. Dr. Fatimi actually shared that she came to this meeting a year or so prior, and it was discussed at this United Leukodystrophy Foundation meeting. And then the structure of DARS2, the gene itself, was actually not really figured out until 2005, but then it wasn't until 2007 until the clinical side of LBSL and the gene DARS2 got connected and were described as being a condition that is caused by changes in this gene. So sometimes we clinically know something first, and then the genetics kind of comes later and things get sort of pieced together, and that's really what happened in LBSL. And the way that it was actually done was through what was called linkage studies in 6 affected sibling pairs, where they were able to kind of use their changes in their DNA. That's kind of what this picture is showing, an illustration of a linkage study, where they're able to kind of narrow down shared regions between the individuals that have the condition and figure out which conditions are then the shared ones that are ultimately only present in the patients with disease. And so that's how they figured out that DARS2 was what caused LBSL. So they've narrowed it down to chromosome 1 first, which is sort of the home that our genetics live on, and then they were sequenced 16 genes that were within that region on chromosome 1, and were able to find genetic changes in all of the patients in that secondary study had changes in the DARS2 gene. So genotype phenotype, so this is the term that we use to describe, can the change in the gene predict clinically what we're going to see? And this is always a question from families, right? So what can you tell me about the changes that my child has, and how is that going to make my child either similar or different from the other patients that are out there. And unfortunately with DARS2, we don't have any concrete answers for this yet. One of the things that makes it difficult is that there are more than 60 different mutations present in the patients who have LBSL, and it's a rare condition to begin with. So we don't have, if we had thousands of people who had it and had 60 different mutations, you could see how we would be able to sort people into different buckets and different groups a little more easily. But because we have, 100 or so patients with LBSL and 60 or more mutations, we have almost as many mutations as we do patients. And so sorting them into clinical buckets and differentiation becomes very difficult. Some of the little bit of genotype and phenotype that's been done, it came out of, you can see at the bottom, the reference, the paper in 2014. They tried to do a little bit of this, and they looked at 45 patients who all have, there's this one genetic change that's very common. It's this guy up here at the top. It's one of these, it's called a splice mutation, and we're going to talk a little bit more about what that is, that affects between exon 2 up here and exon 3. So you can see that this goes out, and each one of these different lines is a different genetic variant. And you can see that this spot right here is a hot spot in LBSL. So there are a number of different genetic changes at that spot, but they are all hitting the same spot. And in fact, the most common genetic change is this one right here that I mentioned, this C228 -21 to -20, where there's a deletion of 2 DNA letters, TT, and an insertion of a letter C. And that one is actually present in about 87% of patients who have LBSL. One of their 2 genetic changes is that change. So they looked at that because it's the single most common to try to see if they could use that as a bucket to figure out some genotype phenotype between those patients. And unfortunately, they all have different secondary variants, right? And there was not sort of an easy bucket that was able to come out. And what they could say is that even these patients that all have this one genetic change in common have a very wide spectrum of severity. So you had children all the way from neonatal to adult onset who all kind of had the same variant. And so it didn't pan out to give us a clear genotype phenotype picture. They also tried to look and say, well, here are the second most common genetic changes. And so the patients who had this one that's really common, plus this one or this one, could they figure out any kind of genotype phenotype, right? So they tried to make buckets. And really, in the end, they all had a variable onset, but they did seem like the patients who had this one plus one of these or the other seemed to have a mildly progressive deterioration. It wasn't kind of as pronounced and more less likely to have kind of the infantile, but that is like this significant, it's really the only thing we've had, and it's an area that just needs to be explored and studied more and tracked better, I think, over time. But they're trying to give this information, get it out there. It's just very difficult, I think, right now with the numbers and the variability within LBSL genetically. The other thing to kind of know, Dr. Fatimi had this image up earlier. This is showing you sort of a map of all of the genetic changes, at least up to 2014, that have been identified in the LBSL gene. The blue box, the blue circles are what we refer to as exons, which are the parts of our DNA that actually code for protein and get directly turned into protein. The in-between areas are what we would call an intron. And Dr. Fatimi's video earlier kind of talked about how you get to this step and then all of a sudden some pieces get cut out right before translation happens. That's removing the introns. And in LBSL, there's actually quite a few, you can see, of genetic changes that seem to affect these regions around the intronic areas. So the regions where those pieces of DNA between the exons need to get cut out and removed so that the exons come together to provide the blueprint. And so that's actually, many of you, I see some heads nodding, many of you in this room, your families probably have genetic changes that affect the splice site, or these areas where the splicing is the process that we call, the word that we use to say cut out this intronic region of DNA so that the exons can all come together and line up tightly to provide us our final code to move on for the amino acids. So genetic testing, I wanted to just touch on this briefly. There are some reasons that people will go through genetic testing and kind of lead them down this process. We don't know exactly what their diagnosis is, how to be able to give a family a prognosis, use it to confirm diagnosis, and often so to provide recurrence risk information for other family members. What do we test? Do we send? Because there are a number of different ways that you could test for LBSL. And many of you may have come to this diagnosis in different ways, but we often will consider the clinical presentation of the patient. An atypical presentation might mean we might send a broader test, and I'll talk about the different options that we have. And then also, the broader the net you throw, or the more genes you look at in any one test, the more likely you are to get uncertain results. And so sometimes the parent's level with uncertainty versus clarity will play into your approach to testing as well. So for DARS2, there are currently, I would say, 3 ways that you could probably find yourself sitting here with a genetic diagnosis of this. One would be you had clinical symptoms, had an MRI, and a doctor very astutely said, I think you have LBSL. Right? And in that situation, those doctors are going to send targeted sequencing of the DARS2 gene. So if everything about you seems like it is consistent with that diagnosis, meaning there's nothing atypical about that presentation, they're going to send targeted sequencing just of that gene. That's important to include deletion and duplication analysis. So earlier I mentioned that we can have misspellings in the DNA as well as extra and missing pieces of the DNA. Very important. Diagnoses can be missed or confused if you don't include that deletion or duplication analysis, as we have learned very well here over the years early on. Turnaround time these days takes about 2 to 6 weeks, depending on what lab you use for DARS2 targeted sequencing. Panel testing. So if you're a little bit unclear, maybe the diagnosis is uncertain, maybe you didn't get to a neurologist who was able to say, I think this is LBSL, they may send a panel. So especially a patient who probably has a lactic acidosis, some doctors, some neurologists may send a broader panel looking at the genes that we know can impact mitochondrial function. And so there are panels that look at 100, 120 genes or so, looking to see all of these different genes that affect the mitochondria and the way they work. And DARS2 is a part of several of those. Panels will take about 10 to 14 weeks to get results back. And then some families, probably not as many, but if we go through some of these first steps and we really can't find an answer, some hospitals are using exome sequencing, and I felt it was important to mention that term for you guys. Exome sequencing is throwing a broad net, basically saying, go find all of the genetic changes in this child, and then let's work backwards to see which ones match up or fit clinically with what's going on with the patient in front of me. So you go in with sort of an unblinded approach of what you're looking for, and you see what comes out and what matches with your patient. Exome sequencing is still a relatively young test, maybe 5 years now that we've been able to use it clinically. So some of you, and as we go into the future, there very well might be patients who present or made with a diagnosis of LBSL through exome sequencing. About the same turnaround time, 8 to 12 weeks. Obviously costs more, kind of each of these things is going to cost a little more. Some considerations with genetic testing, we always talk about genetic discrimination, the idea that could somebody use a genetic test result against you in a negative way. Obviously, when you're trying to make a diagnosis for a child, sometimes the health implications or health risks of knowing what that is trump, right, some of these downstream potential risks. But there is a law called GINA here in the United States that provides protections for group health plans and employment. Insurance coverage, obviously it's important. These tests are not cheap, so making sure it's checked individually for your insurance by your providers. But most labs do have really good financial support that they can help families if they have a big copay or co-insurance for testing. Results of genetic testing can come back in 3 categories: positive, negative, or a variant of uncertain significance. The first 2 are kind of self-explanatory. The variants of uncertain significance are maybe a newer term, so some of you who have had your diagnoses for a little while may not have heard that. It basically means there is a change in the DNA, but there's not enough evidence to say for certain, is this change disease-causing or not? And when you're dealing with rare diseases, a lot of these changes might be novel, might not have been described before. And so your child, even though they have clinical symptoms of LBSL, might be the first one to have one of these changes in this particular gene. And so in that case, a lab may call it as a variant of uncertain significance. clinically, the doctors often, we will look at that and we will be like, I don't care that the lab is calling this an uncertain result. We can predict from what this should be doing to the gene. We can predict from what we're seeing clinically. This is our answer. But it is a term that some of you may have seen on your genetic testing reports, so I wanted to address it. Not all genetic changes impact the function of a gene, and that's important to keep in mind. It depends on a number of different things. Where the change might occur within the gene, intronic, exonic, functionally important parts of the gene, how significant or big of a deal is that particular genetic change. That's my block picture in the right corner. So basically amino acids, those amino acids have different properties to them. Some are charged in different ways, some have different shapes, some like water, some don't. So they have things about them that make them unique. And when you have a genetic change, sometimes it might change it to another amino acid that actually is pretty similar. So if you're supposed to have a 3x3 triangle and it changes it to a 2x2 triangle, you can still wiggle it in the hole that it's supposed to go in your shape, right? But if it changes it from that triangle to a big old hunkin' square, you're not going to get it where it needs to go, and that's going to have a bigger impact on the function of the gene in the end. So there are things that we look at to try to help us predict if a genetic change will cause disease or impact the gene function or not. And also how tolerant that spot is to change over time. So believe it or not, monkeys, zebrafish, they all have DARS2 genes too. And we can look back and see what their order and DNA code is of those genes to know if they're tolerant to change or not at certain spots. So there are official classifications that came out in 2015 for classifying genetic variants. Just to be important, people who were tested prior to this, some of this language is very foreign to you. People who have been tested post this, some of this language is very familiar, and this is when the concept of this variance of uncertain significance really started to come out after 2015. So the last part I want to go through is what some of these different mutations are. And I get this question a lot clinically, people want to understand sort of what their genetic change means, what this change in the DNA, how it actually impacts the gene, and what are these splice sites and frameshifts and nonsense, different types of mutations. So I have a very dear friend, Dr. Iris Gonzalez, who is now retired but used to work at Nemours in the molecular DNA diagnostic lab. And they are actually one of the few labs in the US that does DARS2 testing, coincidentally. I know her through my work with another rare condition called Barth syndrome. And she is the one who developed this analogy. And it's just, I think it's a fantastic way to think about these things. So I want to share it with you today. So the idea is that we're telling you the story of a sweater gene. And basically, the sweater gene provides our instructions that tells our bodies how to make a sweater. And it gives you the list of all the materials. It gives you, and it basically is going to tell you the steps of how to build this sweater. Okay. So basically, the sweater gene is giving you your DNA letters or your blueprint and instructions. Some changes in DNA are called synonymous. That's the term we use when basically the change doesn't actually make a change. Right? So it's a misspelling in the DNA, but it doesn't actually end up changing the amino acid. An example of that, these are actually in the corner here, I've listed out genetic changes that have been reported in the DARS2 gene from a variant database that we all have access to. So you can see here that at position 81, where there should be a T, there is a C in its place. And what it ends up doing is basically where there should be a glycine amino acid, at position 27, it changes it to a glycine amino acid. So it doesn't actually make a change. And that has to do with some of the redundancy in that amino acid code wheel that I showed you, right? So someone who has this genetic variant in their DNA, and then even if they have another one, right, they're not going to have LBSL. This is a genetic change that wouldn't actually cause disease. Compared to a missense variant, and in some situations, a missense variant might actually cause disease and might be a big deal, and in other situations, it might not. So a missense variant is basically the term that we refer to for a misspelling in the DNA code. So a single letter gets switched for another. And in this situation, basically, you can see our example is that at position 580, and that's basically 580 DNA letters into the gene to kind of orient you to the numbers I'm using, where there should be a G, there's an A in its place, right? And what that ends up doing to the gene is where it should make a valine amino acid at position 194. And if we have people that are good in math, you can tell that these are usually, this number and this number are usually divisible by 3 because there are 3 DNA letters in every codon that codes for an amino acid, right? Bringing it all back to what we've already thrown at you for your biology lesson at the end, right? And what it changes it from is a valine to an isoleucine. And the thing to know is that some amino acids, as I said before, have very similar properties. So in this situation of the analogy, what this is doing is it is changing the sweater. It's put a patch on it, right? But that sweater is still very functional, very usable, and isn't necessarily going to cause too much of a problem, okay? You can also have a missense variant where it would change it to a different amino acid that is a much bigger deal. So in this situation at 555, G to a T takes it from a cysteine to a phenylalanine, which have very different chemical properties to it. And in this situation of what's happening to our sweater is you're ending up with a really tiny neck opening that would be incredibly uncomfortable and difficult to use, and making that sweater essentially useless or not helpful, right? We also have something called nonsense variants, which basically will cause a stop in the DNA. And so what we have here is basically 787, you have a C to a T. Again, these kind of look very similar right at the start at the DNA level, but what they do to the protein is the big part of things. What this does is where there should be an arginine at 263, it ends up causing a premature termination or a stop. If that stop happens early in the gene, you're going to end up with only part of a sweater, right? And you're not going to have a complete sweater. If it happens later in the gene, you might end up with more of a sweater, but not as much. In this case, this one happens in exon 13, which is further down the line. The first one happened in exon 9, so you end up with more or less of the sweater created before it causes a stop. Splice variants, which are probably the biggest, right? Because these are the single most common variants that we see, are splice variants and frameshift variants. So in splice variants, basically we lose those signals. So there are signals that tell our body to cut out those regions of DNA that end up bringing the exons all together. And it leads to an incorrect spelling of the exons. And so in this case, you have a splice variant, and so what happens is it ends up leaving extra DNA in place where it shouldn't be. And so you end up with a whole bunch of sort of extra material or extra fabric within the DNA that ends up in the protein, right? So the protein has an extra piece to it and isn't able to work or function quite as well as it needs to. And then frameshift variants, which again are a very common cause in LBSL-2. And in frameshift variants, you have small extra or missing pieces of DNA, usually 1 or 2 letters, sometimes a combination. The most common change is actually a deletion of 2 DNA letters and then an insertion of an extra different letter, right? That 220 that I mentioned earlier, that 87% of patients have, is one of these frameshift variants. And what happens in a frameshift is that you basically start reading through the DNA and then you get to the beginning of the sleeve, and now you have these extra letters that have been added in or taken out, and it kind of derails it, and it starts to make something completely differently, because we talked about the specificity of the 3 DNA letters, tell it what amino acid to put on there, and it basically starts telling it to put different amino acids on there instead of the ones that are supposed to be there. And another way to think about this that I think is helpful is using the idea of a frameshift as a sentence. And this is essentially telling you that basically if we were looking at DNA, these would be groups of 3 letters, A, G, T's, or C's, but we're going to look at it as if we're using a sentence. And basically, if we're going to insert a frameshift right here, right, where we're going to take out the S that's supposed to be there, you can see all of these, what then happens to the subsequent sentence after that. It ends up shifting it over so that the DNA code no longer makes a whole lot of sense. It's still making something, right? It's not necessarily stopped or stopped right away. It still makes something, but it doesn't make Weensy spider went up the water spout, right? So it makes something different. And then often what will happen in these situations is you'll get to a point where it kind of runs out of what's supposed to be there, or it will prematurely code for a stop. And so you'll have sort of an incorrectly made protein that will end up being incomplete. So that is Genetics 101 and then some, right? Because that is clearly not the basics when you get into frame shifts and stop mutations. But I'd like to thank all of my amazing patients and colleagues. And I don't know if I have a ton of time, but I will be around for the expert sessions and stuff as well. We have a couple minutes, so I guess I have 2 minutes if anybody has any questions.
I have a question.
Yes.
Can genes be turned on and off? So, like, I fear that my daughter, who is just has one bad copy, like, this is just my crazy brain, but like, she get a really bad virus or something, and then the other gene gets turned on, and then she becomes symptomatic.
Yeah, so for the most part, our genetics are basically have been there and will always be stable since we were a twinkle in our parents' eyes. So our DNA code is basically formed at the time when the egg and the sperm pass their genes on to become the next generation. One of the few exceptions to that is you can develop genetic changes in things like cancer cells and things like that, but the inherent blueprint of DNA that is in all of our cells of our body does not necessarily change over time. What can change, and what is, I think, part of the research is understanding sort of the spectrum of presentation and why that changes, is how active different genes are in different parts of your body and across your lifetime changes. And so we are obviously, one set of genes need to be really active when we're a fetus in our mother's womb, creating a little tiny human. And then once that baby is born, those genes are not needed as much and different genes become more important. So genes get kind of turned on and turned off, become more active or less active throughout our lifetimes at different parts. And so that might be part of sort of understanding some of the spectrum of presentation and differences there. But the inherent blueprint that is there is what is there and won't change. Right? Okay.
Sorry.
Yep.
So what I heard from your presentation is that we have not been able to tell yet if your specific type variant determines how aggressive the disease is.
Correct.
So if there's somebody in the room who has a patient, a loved one who is very ill, we're not sure if their variant, the type of variant they has anything to do with that. Correct. Okay.
Correct. There has not been any kind of concrete genotype-phenotype correlation that's come out to say that only the patients who have one of these splice site variants and then another variant that takes out a whole lot of function are the reason that this patient is more severe. I think we can theorize that maybe some types of changes might have a little more residual protein function, right? We talked about how Dr. Fatimi said earlier that the mouse that get the gene completely taken out will pass away, right? So that having absolutely no function of DARS2 is not a good thing. And so we do believe that many of the patients probably have some residual function, but it doesn't necessarily hold as concretely to the genetic changes that we're seeing and being able to predict them. It could be just a matter of numbers, and it really could be that with time we'll have a better ability to kind of create those clinical buckets to sort of sift between them. But for right now, it's hard to say. Yeah. Recessive. Yeah, so that.
Does it come from the mother, the father?
Yeah, so typically autism or recessive, each of the parents will carry one change, right? So mom might have a genetic change, and then the other copy of their gene has normal function, so they don't have LBSL. And then the dad may also have a genetic change, and then another one that has normal function, and then the child gets the double dose is what's happening. In some situations, if families have been tested, where the parents have been tested, we may find that one of the parents is not a carrier, and that's possible that second genetic change could sometimes happen new. But I think most of the families, because being a carrier doesn't cause any health implications and you never know that you're a carrier, right, until you have a child with the disease, many of the families, I think, in this room, probably both parents are carriers.
And I have to say, I felt, this sounds bad, but like relieved by that, because to be the one to carry that burden alone, and that is in some diseases the case, I think that's very hard. So we share the burden, I guess, like the emotional burden. And that, it doesn't sound right, but it kind of helps.
To have it be something that genetically comes from both sides makes it a little easier. Yeah. Okay. Thank you, guys.